We will start with Ching-Lung Su. He's an assistant research fellow. He started his own lab at Academia Sinica in Taiwan. He used to work at the Janelia Research Campus, and he's enthusiastic about understanding how computations of neural circuits during behavior arise as a consequence of cellular and biophysical properties of individual neurons. So I will leave the floor to him, and if you have questions, please use the Q&A box, and I will read the question to the speaker. So please, thank you.
Hi everyone, my name is Ching-Lung Shi, and I'm an assistant research fellow at Academia Sinica in Taiwan. So today I'm very excited to briefly share two stories with you about cellular mechanisms for rapidly dynamic neural code in spatial navigation. I've been working on neurons for years. Even so, when I have a chance to take a step back and just look one neuron that I stem, I still feel stunned by the complexity that a single neuron can offer. Besides the morphology, there's a great variety of different ion channels with biophysical properties and their spatial gradients. And there are synaptic connectivity divided by local microcircuit architecture, molecular machineries, and so on. So one can hardly look at this complexity without asking what are they doing there. So as a neuroscientist, I'm totally fascinated by this question. And the question is whether there is some sort of causal link between the complexity of single neural integration and computation and a complex and dynamic circuit function during behavior. I'll put it in another way, how the properties of single neurons can explain the complex circuit function. So I'm particularly interested in the brain region, the dorsal part of the hippocampus. Hippocampus is very important for memory and spatial navigation. And these two functional aspects might be related as the memory being proposed as a form of navigation in abstract mental space. The idea of hippocampus as a spatial navigation system was born out of the discovery of a place cell. Here I show you a spatial firing map of a typical place cell, where the cell only fired more when the animal was near the bottom right corner of this oval-shaped environment. So that place is called a place field for this particular cell. For most of the hippocampal pyramidal neurons, or a given environment, we know most of the cells are silent cells, and only a relatively small fraction of them are active place cells. However, these two functional identities are actually can rapidly interconvertible. So a very elegant study done by Alberto's group a few years ago, they found that when they has an intracellular coding using patch clamp from hippocampal CA1 region, when they identify a single pyramidal neurons, which is a silent cell, they can inject current to the soma to produce a very small depolarization by just a few millivolts. They rapidly convert a function of a silent cell to an active place cell. The opposite was also true when they start with a native place cell. They can convert it to a silent cell by just a few millivolts small somatic hyperpolarization. So, but you might say, well, this is not surprising if that constant current just pushing the overall synaptic depolarization profile above or below the action potential threshold. But the most remarkable finding in the studies, they found that not only the spike is highly sensitive to the voltage, but the subthreshold membrane potential, the activities also have a very nonlinear dependence on the somatic membrane potential. So there must be some sort of magic ingredient here. So when you walk away, I hope I can deliver a view that a nonlinear single neuron property are very well positioned to mediate this kind of type of highly nonlinear response during behavior. We could possibly serve as a putative biological substrate for some sort of context-dependent neural representation. So the first story is the cellular mechanism for the phenomenon that I described, and the gist of the idea is that I can recapitulate or reproduce very quantitatively the phenomenon that observed in vivo by using acute brain slices, wholesale recording from the soma and weakly activating distal synapses with the stimulation pattern that mimic when an animal passes through a place field for a place cell over a time course of seconds long. And the parameter they can use to describe this highly sensitive response baseline VN relationship has a parameter that's nearly identical to what's observed in vivo, including the voltage sensitivity and the amplification ratio, etc. So I can use this strategy to perform very detailed mechanistic analysis, including pharmacology and biophysical analysis, such as compare the amplification mechanism in the soma versus in the dendrites. And as well as computational modeling, which I can quantitatively reproduce the amplification effect observed in vivo using biophysically realistic channel properties. So I can finally test the idea that's or the hypothesis generated by these studies, and then use a half-fixed animal that perform virtual navigation. So once the animal learn to navigate in this oval-shaped environment that mimic the study that I just described earlier, I can combine with whole cell recording the dorsal hippocampus, current injection to single cell as well as single cell pharmacology to derive this picture where the most of hippocampal neuron probably receive some sort of spatial information through the synaptic inputs, but they are mostly weak. So most of the cell have very flat or nearly non-spatial tuning according to animal's position. But if there are some sort of behavioral context or a task engagement, they can deliver this very slow modulation to the system. They can, the modulator of single neuron excitability could engage this substratial gating of the sodium channel near the initial part of the axon. They can dramatically amplify the second long synaptic response, and therefore, like a toggle switch, turn on and off the neural representation of space. So now I only talk about this sodium channel property near the initial part of the axon, but we know there are all sorts of very rich diversity of ion channel and biophysical properties with a spatial gradient. If we put an intracellular recording in the dendrites, we can see this kind of all or none or action potential-like events that can initiate in the dendrites and propagate to a certain part of the cell called dendritic spike, as demonstrated by this biophysically detailed computational model that I established and constrained by many of my experimental data. So the idea, the line of thinking of the function of nonlinear local computation is not only they participate in this moment-to-moment input-output transformation, but also due to their spatial proximity to the input, they are very effectively engaging signaling pathway. They can produce long-lasting synaptic modification or synaptic plasticity by some sort of dendritic nonlinearity like dendritic calcium spike or plateau potential. So the second story is whether this dendritic nonlinearity can produce or support this context-dependent, highly nonlinear neural representation for the space. So consider this scenario. This is a picture of a corridor in Genelia. And on the two sides of the hallway, there are two bathrooms for different genders. But just 50 or 20 meters away, there are another hallway that look exactly identical, except that the spatial arrangement for this bathroom are exactly the opposite. So after a few mistakes, when I just started at Genelia, I realized I really have to learn some sort of context-dependent navigation strategy, which probably means I have to remember where I just came from before I enter the hallway, or look at a subtle size on the wall to know where to get to. So the question is, what kinds of cellular mechanism can support this context-dependent navigation, and what underlies the learning component? What support the learning component at a cellular level to acquire this context dependence? So we can realize this is a realization task in our experiment where the animal was trained to navigate a virtual T-maze. It recognized initial cues and passed through a region where there's no informative sensory or visual cues, and they have to turn left or right to collect the shooter water reward according to the initial cues. Once the animal learned the task, it looks like this. It has to turn to the side direction where the side of the wall have the vertical stripe in the initial part of the maze. And in the delay, in the middle delay region where there's no informative cues, because of it's a VR, we can strictly control the kind of information the mouse can see. So for any differential neural activity observed for that region, it cannot simply be explained by a gross difference of the sensory inputs or the visual input in this case. So this kind of spatial information has to exist somewhere in the brain. 20 years ago, from a few very beautiful study, we know there are place field firing can strongly be modulated by the animal's travel trajectory. So therefore, whether the place cell can fire for or express its place field depending on whether the animal turns to the direction the animal turned in a given task, which is some sort of choice-related place cell firing. So the question here is whether the local nonlinear events were able to support this kind of very rapid and reversible context-dependent neural representation. So the short answer is yes. Once the animal learned the task, I start to patch a neuron from a dorsal CA1, and if the CA1 pyramidal cell is a silent cell, I deliver a very short current pulse for an animal pass through a fixed region during the delay, but only applied to one trial type. And to treat this very large and short current pulses can trigger the calcium plateau potentials. If we start from a silent cell like the example shown here and only trigger the plateau potential in one trial type like the right turn trial here, but not the left turn trial, it can immediately induce just within a few seconds a very robust place field firing that lasts throughout the recording session, but only in the trials in which the plateau potential was triggered, but not the other trial type. If we look at the firing frequency plotted as a function of animal's position, we see the firing field induced during the induction site has a very dramatic difference between the two trial types. If we look at the intracellular membrane potential, the synaptic depolarization also shows a very dramatic or very remarkable difference between the two trial types near the induction site after the induction. So for the sake of time, I just want to give a very, very high level and very short summary, which is the effect of nonlinear local integration here seems to be drive the potentiation of synapse and therefore the support a manifestation of the bioselectivity to the combination of initial cue and the place. And what I don't have time to cover here is that the specific task engagement driven by learning seems to play an important role to determine whether the initial cues are meaningful context for this system. So with that, I want to thank my previous advisor, Nelson Spruceton and Xinyu Zhao, who I designed all the behavioral tasks with together. And finally, I want to end by saying that my lab is recruiting academia in Taiwan, academia Sinica in Taiwan. So welcome everyone is interested. Thank you.
Thank you very much, Ching-Lung, for the wonderful talk and very nice experiments. While the question and from the audience can arrive, I will say that it's impressive that also in the virtual reality setting, the mouse is able to learn this not so easy task, not so easy also for us human as you mentioned. So really impressive work. And so we have one question. I will read it to him. So from Aditya, do you see any EC CA3 input differences for context-dependent inputs?
ECCA3 input. What's the question again? Could you repeat?
So if you do you see any EC/CA3 input differences for context-dependent inputs?
Yeah, that's a very interesting question. I mean, for now we only record from the soma, and there's no control of which input we are actually looking at in the in vivo condition. But I guess part of the one way to approach that question is really a fundamental question of how the plateau potential plasticity rule work, like how much or how specific they're to a particular pathway, and even the inhibitory circuits, microcircuits. So that's actually one of a focus of one of the project I want to start on in my lab. But the general idea from older work is that the seems to CA3 might be greatly potentiated by the plateau potential, but I think the evidence is more indirect.
Perfect. Thank you very much for answering the question and for your talk. I think it's time to move on to the